So we start our microbiology journey in the GI tract with the title Natural Defenses of the GI, as well as the microbial populations that are normally resident of the GI tract. So our lecture will include two parts. We will talk in the first part about an overview of the immune responses in the gastrointestinal tract and their distinctive feature. And in the second part of the lecture, we will be talking about microbiome, or what used to be called the normal flora. So let us start with some key facts regarding the gastrointestinal tract. Firstly, the GI tract has the largest surface area in the body. It's an enormous area that needs to be protected. It has a surface area of almost 400 square meter, which is 200 times that of the skin. Secondly, we have the microbiome. At the same time, the healthy large intestine is colonized by at least 1,000 species of bacteria that live in symbiosis with their host and are known as commensal microorganisms, what we will be calling from now on as microbiome. Therefore, the GI tract requires a continuous protection during ingestion of food and water from all infectious and non-infectious dangers, as well as preventing any microbial breach or imbalance caused by the resident microbiome. Regarding affected patients with gastrointestinal disorders, diarrhea is still the most common presenting symptom of gastrointestinal tract infections and remains a leading cause of mortality and morbidity worldwide, especially in children. However, fortunately, it is both treatable and preventable. For the GI protection, there is naturally an anatomical and physiological barriers that, of course, include the skin, the oral mucosa, and the intestinal epithelium, where the cells are joined by tight adhesion junctions, as well as the chemical barriers that include the stomach acidity, which is unfavorable for most microbes. This would be followed by the activation of the innate immune system, namely the complement system, the antimicrobial enzymes, the antimicrobial peptides, as well as the secretory molecules like the secretory phospholipase A, the mucus, as well as the secretory immunoglobulin A, which is the predominant class of antibody in the mucosal immune system, and it is produced locally by plasma cells in the mucosal wall. So protection against pathogen rely on several levels of defense. The first line of defense against invasion by potential pathogens and commensal microorganisms is the thin layer of epithelium that cover all these surfaces. However, the epithelium can be breached relatively easily, so its barrier function needs to be supplemented by defenses provided by cells and molecules of the mucosal immune system. Secondly, various chemical and enzyme systems, including the complement, as we mentioned before, act as an immediate antimicrobial barrier near these epithelia. If epithelia are breached, nearby various innate lymphoid cells can coordinate a rapid cell-mediated defense. If the pathogen overcomes this barrier, the slower acting defense of the adaptive immune system are brought to bear. So cell-mediated immunity proceeds in a series of steps. We have the inflammatory inducers, which are chemical structures that indicate the presence of an invading microbes, and we refer to it in immunology as BAMPs, pathogen-associated molecular patterns, or the cellular damage that produced by those microbes, and we refer to them as DAMPs, damage-associated molecular patterns. On the other hand, the sensor cells detect these inducers by expressing various innate recognition receptors. We call them pattern recognition receptors. And the response they produce a variety of mediators that act directly in defense or that further propagate the immune response. And the mediators, they include many cytokines. They act on various target tissues, such as the epithelial cells, to induce antimicrobial proteins, antimicrobial peptides, and they resist the intracellular viral growth. Or on other immune cells, such as the innate lymphoid cells that produce other cytokines that will amplify the immune response. So let us start talking about the epithelial surface of the body that provide the first layer against infection. So on the microenvironmental level, the epithelial surfaces provide the first layer of protection, where the surface area available for nutrient absorption is maximized by a finger-like protrusion. We call it the villi, which are predominantly covered by a columnar epithelial cell to maximize absorption. As well as those epithelial cells, they are known as the intestinal epithelial cells, or collectively as enterocytes. Between the villi, we have the crypts of Leperkuhn, and these crypts include the mucus-producing goblet cells, as well as the stem cells, which give rise to all intestinal epithelial cell lineages. These crypts include, as we said, the mucus-producing goblet cells. The mucus play a key role in protection against infection in the gastrointestinal tract, as well as we have the Paneth cells that located at the base of the small intestinal crypts, where they secrete antimicrobial molecules. The forest of villi is interrupted occasionally by lymphoid nodules, referred to as Peyer's patches. As we could see in the left to this picture, Peyer's patches, they include a specialized type of epithelial cells. They are called the microfold cells, or M cells, which are specialized enterocytes that allow the luminal content to be passed through and encounter an antigen-presenting cells below. However, this feature has been hijacked by some pathogens, and this M cells serve as a point of entry for pathogens like Salmonella enterica or Shigella or Yersinia pestis. We have the lymphocytes and other immune cells such as the macrophages, the dendritic cells, that are found through the intestinal tract, both in organized tissue and scattered through the surface epithelium of the mucosa, and the underlying layer of connective tissue, what we call the lamina propria. The abundant intraepithelial lymphocytes present in the intestine, more than 90% of the intraepithelial lymphocytes are in the small intestine, where they are mainly T cells, and as you can see, they are mainly CD8 T cells, and that's in complete contrast to the lymphocytes that are present in the lamina propria, where they are predominantly B cells. So the mucosal immune system is something different and has distinctive feature from the systemic immune system. While most internal tissues are free of active microbial growth, the skin and the various mucosa lining organs that directly contact the external world will have a continuous encounter with the environmental microbes. These surfaces are where most pathogens invade. The mucosal immune system, in particular that of the gut, may well have been the first part of the vertebrate adaptive immune system to evolve, possibly linked to the need to deal with the vast population of commensal bacteria that co-evolved with the vertebrates. Those organized mucosal surfaces have specialized immune structures that orchestrate the response to the environmental microbes encounters, and include, as you could see in the picture, the GI tract, the upper and lower respiratory tract, the urogenital tract, as well as you have the middle ear. It also includes the exocrine gland associated with these organs, such as the conjunctiva, the lacrimal glands of the eye, the salivary glands, as well as the lactating breast. So in contrast to the systemic immune system, the mucosal immune system is bigger, encounter a wider range of antigens, and encounter them much more frequently than the rest of the immune system, what we called the systemic immune system. This is reflected in a distinctive anatomical features as well as specialized mechanisms for uptake and unusual effector and regulatory response that are designed to prevent unwanted immune response to food and other onocous antigens. That we will cover some of those distinctive features in the next slides. So we start with the mucosa-associated lymphoid tissue, and specifically in the GI tract, what we call the gut-associated lymphoid tissue. The gut comprise the Peyer's patches, which are present only in the small intestine, as well as we have the isolated lymphoid follicles, which are found throughout the intestine. We have the appendix, as well as the tonsils, the adenoids in the throat, as well as the palatine tonsil, the adenoids, and the lingual tonsils that are large aggregates of lymphoid tissue covered by a layer of squamous epithelium and form a ring, what we know as Waldeyer ring, at the back of the mouth around the entrance of the gut and the airway. So the internal mucosa of the small intestine is made up of a finger-like projections, what we called the villi, that covered by a thin layer of epithelial cells that are responsible for digestion and absorption of nutrients. The tissue layer underlying the epithelium is called the lamina propria, as we mentioned in a previous slide, and the lymphocytes are found in several discrete compartments in the intestine with the organized lymphoid tissue, such as the Peyer's patches and isolated lymphoid follicles throughout the intestine, forming what's known as the gut-associated lymphoid tissue. These tissue lie in the wall of the intestine itself, separated from the content or the content of the intestinal lumen by a single layer of the epithelium. Of course, with the draining lymph node for the gut, which is the mesenteric lymph node, as well as the caudal lymph node, which are connected to Peyer's patches by afferent lymphatic vessels, and they are the largest lymph node in the body. Together, these organized tissue are the sites for antigen presentation for T and B cells and responsible for the induction phase of the immune response. If we take a closer look to the Peyer's patches, they are important sites for initiation of immune responses in the gut. They are visible to the naked eye, where they have a distinctive appearance, forming a dome-like aggregate of lymphoid cells that project into the intestinal lumen. Peyer's patches and the mesenteric lymph node, as we said, they contain a discrete T cell areas. You could see them here in brown, and predominantly B cells in those follicles in the blue. So the main lymphocyte type in the isolated follicles is B cells. The intestinal mucus is playing a key role, a key protective barrier in the gut. It's an efficient system for protecting the epithelium from the bacteria by promoting their clearance and separating them from the epithelial cells like a buffer area, thereby inhibiting the inflammation and infection. So the function of the colon inner mucus layer is especially important, as this explains how we can harbor such a large number of bacteria in our gut. And of course, the major component of this mucus system is the mucin type 2, which is organized the mucus by its enormously large net-like polymers. And the pathogenic microorganisms in turn have developed mechanisms for circumventing this well-organized mucus protective system. Mucus has another crucial role as well. It holds up the immunoglobulin type A at the epithelial surface of the gut, and we know that immunoglobulin type A is the predominant antibody class in the gastrointestinal tract. Another distinctive feature of the mucosal immune system in the gastrointestinal tract is the uptake and transport of antigens by M cells. So antigen present at the mucosal surfaces must be transported across an epithelial layer from the lumen, so can be presented to the antigen presenting cells before they can stimulate the mucosal immune system. Thus the intestine has a distinctive routes and mechanism of antigen uptake. This panel illustrates the passage of antigen through the M cells. We talked about those microfold cells in the follicle-associated epithelium or Peyer's patches. Those M cells have a convoluted basal membrane that form pockets within the epithelial layer, allowing close contacts with lymphocytes and other immune cells. This favors the local transport of antigens that have been taken up from the intestine by the M cells and their delivery to dendritic cells and other immune cells for antigen presentation, and material transported through the interior of the cell when they cross the M cells in a membrane-bound vesicle to the basal membrane, where it is released into the extracellular space in a process known as transcytosis. On the other hand, you have the immunoglobulin that is secreted from the gut and need to reach the lumen, the gut lumen. And we know that the predominant class of antibody in the gastrointestinal tract is the immunoglobulin type A, and the nature of the immunoglobulin type A differ between the two main compartments in which it is found. You have in the blood, it is found as monomers, and in the mucosal secretion, it is as a polymer or usually as a dimer, and the two immunoglobulin monomers, they are linked by a J chain to reach its target antigen in the gut lumen. The IgA has to be transported across the epithelium by the polymeric immunoglobulin receptor. The bound complex undergo transcytosis, by which it transported in a vesicle across the cell to the apical surface. There the polymeric immunoglobulin receptor is cleaved, leaving the extracellular immunoglobulin A binding component bound to immunoglobulin A molecule as the so-called secretory component. Part of the cleaved polymeric immunoglobulin receptor remain associated with the immunoglobulin type A and is known as the secretory component. The resulting antibody is protected from proteolytic cleavage, and it is referred to as secretory immunoglobulin type A. And then the secreted immunoglobulin type A can bind and neutralize pathogens and toxins in the gut lumen. In the second part of the lecture, we will be talking briefly about microbiome, or as I mentioned before, what we use to be called normal flora. The term normal microbial flora denotes the population of microorganisms that inhabit the skin and the mucous membranes of healthy normal persons. The microorganisms that live inside and on humans, now referred to as the microbiome, they are also called commensal microorganisms, since they can have a symbiotic relationship with the host that consists of archaea, bacteria, fungi, but cause no damage to the host, And in some lectures, it's a mutualistic relationship. It's beneficial for both the parasite and the host. We humans are mostly microbes. We have over more than 100 trillion of those microbes or microbial cells, and they outnumber our human cells by the ratio 10 to 1. And the majority of them live in our gut, and the more distal you go, the larger microbial population you will find, particularly in the large intestine. Even with these enormous numbers of microbes in the intestinal microbiome, they normally cause no damage and are confined within the intestinal lumen by a protective layer of the mucus that play a key role in buffering the bacterial microbiome from being in direct contact with the epithelial layer. And this is different from the pathogenic bacteria that can penetrate this barrier and injure the intestinal epithelial cells and spread into the underlying tissue. Where are they? We could have a look at the pie charts, the different sizes of the pie charts for different sites reflect the number of distinct bacterial species typically present at those sites. The colon contain the greatest number of different species, over 1,000 estimated from individual surveys. The color key indicates the 4 bacteria phyla that contain the majority of the commensal bacteria. The commensal bacteria include the firmicutes, like the lactobacilli and the Clostridium species, as well as you have the actinobacteria, like the bifidobacteria, the commonest inhabitant in newly born babies, as well as you have the bacteroid fragilis, the anaerobes, that are among the common commensals in the colon, as well as you have a population from the proteobacteria like the E. coli or Proteus. The variation in the microbial identity between the samples differ based on where the samples are taken from different body sites. Who are they? As we said, they are mainly firmicutes, bacteroids, as well as the actinobacteria and proteobacteria. And they always, the skin and the mucous membrane harbor a variety of these microorganisms, and usually we divide them into either resident microbiota, which are fixed types of those microorganisms that are regularly found in a given area at a given age, that if disturbed promptly re-establish themselves. And we have, on the other hand, the transient microbiota, which are non-pathogenic. However, they are potentially pathogenic microorganisms that inhabit the skin and the mucous membrane for hours, days, or weeks. This slide explains the types of the commensal bacteria that reside in the human host, and as we said, they include the firmicutes as well as the bacteroids as the commonest commensal bacteria in the gastrointestinal tract. They account for more than 90% of the microbiome in our bodies. We have as well the proteobacteria, like the E. coli, Proteus, Salmonella, as well as the actinobacteria, and the representing species in this class is the Bifidobacterium species. And as we mentioned before, the variation in the microbial identity between the samples taken is different based on different body sites. What can influence the microbiota? The microbiome is dynamic and subject to important changes during the life of the host in response to a variety of factors, including the diet, the environment surrounding the host, any medical intervention, and the disease states. For example, if we take the route of delivery, it can affect the microbiome population. Infants delivered vaginally will harbor microbial communities dominated by the lactobacilli as well as the bifidobacterium species, which are similar to their mother vaginal canal. This is in contrast to infants delivered by caesarean section. Those infants are colonized by microbial communities composed of common skin microbes, and the commonest is the Staph aureus, is the predominant genus. So at birth, the intestine is sterile, but organisms are soon introduced with food. The environment, as we said, the route of delivery, for example, maternal, vaginal, as well as being exposed to fecal or skin microbiota are major factors in determining the early microbial profile. Many early studies reported that the intestinal microbiota of breastfed children is dominated by the bifidobacteria. So bifidobacterium is the commonest commensal bacteria living in infants. Once the intestinal microbiome is established, an orderly developmental program unfolds during which its composition and function capacity converge toward a mature configuration. This process is punctuated by dramatic shifts that correlate with the factors we mentioned before and events and circumstances in early childhood, especially the induction of solid food, for example, for infants during weaning, where microbiome shift from being predominantly bifidobacterium species to other anaerobic bacteria like the Clostridium difficile species. What do the microbiota do for us? The important functions of the intestinal microbiota can be divided into three major categories. The first of these are the protective function, in which the resident bacteria displace and inhibit potential pathogens indirectly by competing for nutrients and receptors, or directly through the production of antimicrobial factors such as bacteriocin and lactic acid. So they fight or compete for space and nutrients. On mucous membranes and the skin, the resident microbiota may prevent colonization by pathogens and possible disease through a process known as bacterial interference. Second, commensal organisms are important for the development and function of the mucosal immune system, and this topic will be covered in the immunology course next year. Indeed, some commensal organisms perform important functions, as in the case of the bacteria that aid cellulose digestion in the stomach of ruminants. The difference between commensal organisms and pathogens lie, as we mentioned before, in whether they induce damage to the cells. There is still much to be investigated regarding the microbiome in a broad attempt to understand the role that is played by the resident microbial ecosystem in the human health and disease. And in 2007, the National Institute of Health in the United States has launched the Human Microbiome Project to investigate the role of microbiota in human health and disease. Microbiome, they are beneficial, however, they can be pathogenic potential under certain circumstances, such as if they are introduced into other sterile body sites, for example, with trauma, when the host immune response become compromised, as well as when there is dysbiosis, and dysbiosis represent an imbalance between a bacterial species in the microbiota after exposure to, for example, antibiotics abuse, and we have the case of Clostridium difficile associated diarrhea, or what is known as the disease pseudomembranous colitis. It is increasingly recognized that the microbiome can change our mind and health status or switch on a wide range of diseases that include cancer, cardiometabolic diseases, allergy, obesity, and other autoimmune diseases as well. Even the gut microbiome has been associated with autism and other mental diseases. Does it have to do with the hygiene hypothesis? In medicine, the hygiene hypothesis states a lack of early childhood exposure to infectious agents or symbiotic microorganisms like those in the microbiome, such as those in the gut, and parasites increase the susceptibility to allergic diseases by suppressing the natural development of the immune system. In particular, the lack of exposure is thought to lead to defects in the establishment of the immune tolerance. This hygiene hypothesis has also been called the microbiome depletion theory or the microbial diversis hypothesis and the lost friends theory. It's a rational explanation for the link between microbial exposure and inflammatory disorders in the future. We mentioned some examples of links between microbiota and disease. The gut microbiome is different between, for example, in obesity, between obese and lean twins. Obese twins have a lower diversity of bacteria and higher levels of enzymes, meaning that obese twins are more efficient at digesting food and harvesting calories. And obesity has also been associated with poor combination of microbes in the gut with increased proportion of the firmicutes. As well as microbiota has been linked to other autoimmune diseases such as the inflammatory bowel disease, rheumatoid arthritis, as well as diabetes type 1, which is an autoimmune disease that is associated with less diverse gut microbiome. And in animal studies, bacteria play a role in developing diabetes. The role of microbiota has been established as well in GI cancers, with as well the common oral diseases like the cavities and gingivitis that are the most common infectious diseases worldwide. A correlating link as well has been documented between the microbiome and the allergy-like diseases. And lastly, the conspicuous example is the pseudomembranous colitis, which refers to a swelling or inflammation of the large intestine, the colon, due to an overgrowth of the Clostridium difficile bacteria. This infection is a common cause of diarrhea after antibiotic use and abuse, and the fecal transplantation has shown to cure patients with pseudomembranous colitis. Can we manipulate the microbiota? Yes. For example, by administering probiotics. Just notice the difference between probiotics and prebiotics. Probiotics are live microorganisms intended to provide health benefits when consumed, generally by improving or restoring the gut flora. Probiotics are considered generally safe to consume, but may cause bacteria-host interaction and unwanted side effects in rare cases. On the other hand, prebiotics are compounds in food that induce the growth or activity of beneficial microorganisms such as bacteria or fungi, as well as drugs that adjust the immune system, such as those used in cancer treatment, as well antibiotic, either excessive use or abuse, and the development of resistance later on in life. The bacteriophage therapy has been used to modulate the microbiota. However, it is noticed that the resistance develop so rapidly. And one of the documented ways to manipulate microbiota has been the fecal microbiota transplantation. The one I mentioned before as being used for treatment and cure of Clostridium difficile diarrhea, or what is known as pseudomembranous colitis. It's a clinical procedure that restores healthy bacteria in the colon by introducing a stool from a healthy donor through colonoscopy or enema. Potentially fatal Clostridium difficile infection have been cured using this fecal transplantation to restore the healthy gut microbiota. Thank you.